Hey, boys and girls and other varieties. We're here to try to learn something about DNA. And David, you are the victim.
Yeah, I am the victim, all right.
You're the victim. So, and because you're the victim, yeah, you're kind of going to kind of steer this with, questions and things that you don't understand and all of that. So this is my hope, because if you just start explaining without realizing what people are missing, then you tend to lose people, and I don't want to do that. So, and I ain't very good at this. I will admit this right off, because I often confuse myself when I start trying to teach something, because it's like, oh, wait a minute, I don't know that part. Anyway, so let's see. I have a picture of some DNA, I believe. Screenshot, let's see, screenshot 2106. And, no, that's not the one. This one. So, I don't know where to start. I think we could start here. Can you see that at all?
Yep.
Is it fairly visible on the screen? Anyway, so, we got the DNA double helix, with you're probably familiar with.
Yep.
Do you have any questions about that?
No, I'm not so far. I'll do my best on questions. I don't know how good I'll do with that, but we'll see.
Okay, we'll get a little further into that. But I think one thing I could do here, I'm going to stop the screen, share the screen, try to get rid of settings, window, and I want to go over here and show this. Okay, so basically, first of all, we have a cell. So big cell, right?
Yeah, that's exactly where I thought we would start. But yep, good.
Okay. Inside the cell, we got big nucleus. And we're talking about eukaryotes now. And I'm not going to talk much about prokaryotes, because that would probably just confuse things. So inside of the cell, we have these chromosomes, which are usually drawn, kind of like, with this sort of X shape thing that you see all the time, right? Like that. But they're not like that normally. They're like that when they get hyper condensed, when the cell divides, or when gametes are produced. So when a cell divides, it's called mitosis. And these things, gather up and they do a bunch of stuff and they try to duplicate themselves as well as possible. And then the cell divides, and that happens all the time in your body. But at certain stages in an organism, we have meiosis, which is where all of the recombination and all the complicated stuff happens that we talk about in evolution. So we'll just table that for now. But in normal operation, they're finding out, they recently found out that cells have areas, like in the nucleus, or not, chromosomes have areas, they have territories, they're called, right? In the nucleus. And okay, say we have chromosome 2 here, and it's sort of, we'll show you how it's folded up, but it's folded and parts of it are unfolded, and, it kind of loops out. And it could have, like a tail sticking out into some other area. And then over here, that's one copy of chromosome 2 from your mama. And then you have another copy from your daddy over here. And it might have its own territory. And recently they found out that these territories overlap and like the chromosome, I think it's 15 or something, might be next to chromosome 14. So there'd be another one. And they found out that in organisms and sometimes even across species, that these territories seem to be, they have some sort of a relationship, but there's a lot we don't know about it yet. But anyway, so the chromosome is basically in a state where it can be transcribed into proteins and into RNAs and things like that inside the cell. So there's and these things seem to have their own little place where they want to be. Now they're trying to figure out right now, so this is just real current state-of-the-art shit, but they're trying to figure out what's going on at the inner surface of the nucleus that allows these things to stick where they do and to accumulate where they do. And this whole thing is being studied in something you'll hear from creationists in the 4D nucleome project. And I think you'll hear that from Otangelo, and you'll hear it from Cell. And but anyway, they're trying to figure out this 4-dimensional, basically organization of chromosomes. So this is like state of the art stuff. So anyway, that's the first thing. So we've got 26 chromosomes. You know, we got what the hell we got? Yeah, 22. You know, that are paired up, and then we have the XY chromosome if you're a male, or the XX if you're a female. So,
Yep.
So you got one of each. You got one from your daddy, one from your mama, and these things are cranking out proteins and RNAs and things like that. And there's a process called transcription that happens in the nucleus, where it goes from the genome or the chromosome, transcription, and then you get an mRNA. Okay, that's a messenger RNA that pops out here, and then from mRNA you'll get a protein. And the whole idea is to get these proteins and various other chemicals in the cell. So, okay.
Yeah.
That is called translation. So, How much of that did you know, or do you know?
None of it.
Oh, shit. Okay.
So yeah.
So anyway, the important thing is we got a nucleus, we got chromosomes in there. They're in some state so that they can be transcribed, or there's transcription, and then from there, messenger RNA is popped out of the nucleus into the ribosomes, which are the factories that do the translation. Okay. Not quite like that, but that's simple. The ribosomes attach to the mRNA and they translate it into proteins. That is kind of the main idea of genomes and chromosomes. So, and then, okay, so you got two strands. We got one strand here, and we got its duplicate, right? So this is chromosome, let's say chromosome 3.
Do you realize you popped off the screen, right? Your thing.
Oh,
You're not sharing.
Yeah, you're not sharing.
Yeah, what just happened? Wow. It crashed. Hang on a second. How long would that happen? Because I wasn't
Only minute, like minute, not even that. 30 seconds.
Huh. Oh, let's see. Do I have a crash warning? I don't know what happened. Put it back up.
Hey, David. Hey, Speed.
Hi,
Andrew.
Andrew. How are you? I'm hoping you're going to help me with my class.
Yeah.
I'll do my best. I don't know everything there is to know about genetics, but I can try.
Yeah, but you certainly know far more than I do.
Who the hell does?
Yeah.
True.
Yeah.
And maybe because I wouldn't say at the beginning, but still in the process of learning, maybe you can help too. Kind of. I don't know.
Okay. So we got chromosome 3 from the mama, and then we got chromosome 3 from the papa, basically. So this is the double strand. They're actually, it's actually that helix, but I'm not showing it as a helix. So basically, the chromosome 3 has a beginning and it has an end. So, and beginning and an end. And so you'll hear 5 prime, 3 prime. We'll explain that a little bit. We got 5 prime, 3 prime, and that pairs up with 5 prime, 3 prime on the other side. So we got daddy's chromosome and we got mama's chromosome. Okay. Now, there's a word that we toss around a whole lot, and that is locus or loci. Loci plural, locus. And so what a locus is in a genome is in some ways sort of whatever in the hell you decide it is. But typically, we're looking for something called a gene. So we got a gene here, and this is going to be the locus of that gene right here, right? So this is like the location. And there's a matching location over on Papa's chromosome. So we have another one. So the gene is... So we have 2 genes that can be transcribed, and these can be homozygous or identical between Papa and Mama, or they can be heterozygous or different. And if they're different, then we say that they are allelic to each other. So it's an allele, meaning allele sort of means it's short for other something or another. It's allelic something. It's something long that they shortened, and they call it an allele. So, and that means that there are some differences somewhere in this gene between mama and daddy. So, okay so far, Andrew. Have I screwed anything up?
No, I don't think so.
Okay. Because I tend to do that. Tend to get lost in my own brain often, too often. Okay.
If I can add something, you may have already said this, but I would just describe an allele as different versions of the same gene.
Yeah. But it could be something else too. And there's a little bit of some people. Okay. There's some. Should we talk about SNPs? Single nucleotide polymorphisms, and what is the other one? Single nucleotide. Shit, lost it. Okay, so anyway, a locus can be any section of the gene that we pick. Now on this one we have I've drawn the gene part, right? This is the part that gets transcribed. Okay. But there's often, a part right before that promotes it. And then there's a couple other parts that will enhance or inhibit it. So we could call the locus this whole thing. And we could talk about that whole thing. So locus is kind of what the area of the genome that you're interested in, that you're going to be talking about. And you're going to say that there are alleles or there are differences either between the 2 genes you have in your cells or between genes that we have in a population. Okay. So there could be I could have exactly the same copy of gene A here and A here. So uppercase A, and out in the population, somebody might have an A, uppercase A, and they might have a little lowercase a. So this gene is different in you than it is in me. Or it could be different in me. I could have 2 copies or 2 different copies of the gene. So this person over here has an A, uppercase A, and a little lowercase a, and I have 2 A's. Okay. So I'm better and faster and stronger because I have 2 A's. Maybe.
So what happens when they're different? I don't know if that's even a question or not. So
when they're different, they could possibly produce a different protein, or when they're different out here. Now this guy is different, right? So he's got an uppercase A and he's got a lowercase a. There's a lot of things that could happen here. It could make no difference at all. It could just be an oddity that we have to track in a population. So it may create a protein that works just as well as uppercase A. On the other hand, it may create a protein that it doesn't work at all. And
right.
if you
I need to interrupt you. Sorry. Look who's here. Demeter is here, so you're in trouble. So you better get this right.
Oh shit!
I'm sorry. I'm sorry. I didn't mean to put pressure on you, but I just.
Yeah. Okay.
Okay.
Single nucleotide polymorphism, and what is let's see, single nucleotide. There's another one. There's another 3-letter thing that I'm trying to find in my brain. Okay, so you may have 2 lowercase a's that don't work. In that case, you die.
Really?
Fucked. Yeah, you die. Well, maybe
it would kill you.
It could kill you. Yeah.
Really?
Yeah.
Okay.
It could kill you. Well, it depends. And in that case, that organism would never reproduce, so we wouldn't have to worry about it, right?
Are you going to talk?
Yeah.
Oh, sorry.
What's that? Okay,
are you going to talk about dominant and recessive alleles in this case?
Yeah, I'm not sure. I'm going to talk a little bit about that, but we don't want to go too far into that.
We don't want to go too far, Andrew. You got to don't forget. We're just starting this, and we probably will continue, right?
Yep.
Maybe with this. This is kind of something that we want to do here.
Okay.
Right. So.
Okay. So it could be that those things don't work at all, and it could be that they work less well, or that they create a different trait or whatever. And there's a whole bunch of combinations between the two, like for sickle cell anemia, the sickle cell thing. If you have 2 A's, you will get sickle cell anemia. And basically, it will interfere with your ability to stay alive. And I don't know much about that disease. Do you know anything about it, Andrew?
Let's see. You're talking about sickle cell anemia, like in terms of malaria and all that.
Yeah. No, no. If you have that A allele, okay, you have that difference. Mosquitoes don't. I can't draw a mosquito. Let's say a little mosquito, and he's got a big, huge spike that he sticks in you, right? So that's why mosquitoes don't like that little A, so they can't attack you, okay? But if you got 2 little A's, you're probably going to be really sick and die. You will develop, and I don't know how the disease plays out in a person's life. I think you can reproduce, can't you? But
I do need to correct you, Speed.
Okay, go ahead.
So it's not the mosquitoes that can't infect you.
Oh, it's malaria?
Yeah, it's the malaria parasite itself, because in the case of sickle cell anemia, the misshapen blood cells as a result of the specific allele combination, it can't attach as well. So it's not the mosquito, it's the malaria parasite.
So in this case, if you have 1 A, you're protected from malaria. If you have 2 A's, you're going to get really sick and have sickle cell anemia. If you have two uppercase A's, you're not going to be protected from malaria. So in populations where malaria is a real problem, this AA thing ends up the amount of lowercase A's and uppercase A's you have, if you counted them across an entire population, would end up about 50-50. It's really interesting how they develop.
So 50-50 would get it, and 50% would get it.
Not like that. If you if I count, this person has 2 uppercase A's. Okay, this one person, one from the papa, one from the mama. Yeah. This person over here has a lowercase and an uppercase.
Yeah.
So if I were to count the uppercase A's, I would have 3, and I would have 1 lowercase A. Okay. Yeah. Because this is Papa, and this is mama here, right? So I have 3 uppercase A's and I have 1 lowercase a. Well, the way it turns out, and again, I haven't really done anything other than just peruse this information. It turns out that the lowercase a and the uppercase a in populations with high malaria, basically threat, will end up with 50-50. It's really interesting, and it sort of works out so that a lot of people end up with this lowercase a, uppercase a combination. They're the ones that survive the best. And it ends up being, of course, there's a distribution, and malaria will still kill them, and sickle cell anemia will kill them, and but for the most part, these guys that have this combination survive rather well. So we probably went a little too far into that, but let's see. So what else? Okay, so we're talking about locus and loci. Okay,
so since we went too far into that, what kind of takeaway do you want us to have
on that?
Or so
that at a locus, you can have genes that vary within an individual. You can have as many as 2 different kinds of genes at that area. You could have 2 A's. You could have or two uppercase A's. Hang on a second. What did I do? Okay, you could have 2 uppercase A's. You could have 2 lowercase A's, or you could have an A and an A. So, okay, and if you have this, it's called heterozygous. So you've heard that term many times.
Yes.
If you have this, it's called homozygous. That means you have the same copy, right?
Yeah, homozygous dominant.
Okay, I gotta remember that. Yeah. Okay.
Okay, so that's I probably should write it, but I'm not gonna. Maybe I will later. Okay, so in this is just one person. This is a person. Okay.
Yep.
Can have one of those three conditions. Okay, but then in a population, like if we look at a population, this guy might have an AA. This guy will have an AA. This guy will have an AA. I like going to AA. I like AA. Okay.
Yeah.
So over the entire population, you have this mix, right? So we could say this guy has an AA. So what you do is you count the amount of this one gene that is identical. Mostly, well, not even going to talk about that. Okay, 1, 2, 3, 4, 5, 6, 7. So we have 7 A's. 7 of those, and we have 1, 2, 3. So we only have 3 of those other ones. Okay, so that is the total. Okay, so there's 10 total alleles. The uppercase A's are 70%, and the lowercase A's are 30%.
And this would be kind of like evenly just distributed throughout the population, or not evenly, but
not evenly. Yeah, it could slide one way or the other. Yeah.
Right. Generally. Okay. Yeah.
Okay. So if you have a big population like human beings, when you look at this one locus, like Dr. Dan mentioned for hemoglobin, I think with one of the hemoglobins, there are 1,000 variations. So you would have to A, B, C, D, A1, B1. You have there's 1,000 variations across our entire population, but one person can only.
Yeah, that was okay. Go ahead, continues.
So
one person can only have. Go ahead,
if I may. Okay, because in certain areas, the geometry or 3D space of it matters a lot. When you have the heterozygous combination, the big A, little a, does it matter which side of it it's on? Like, what if the one on the left was the little a and the one on the right was the big A? Would it behave differently?
They're pretty much interchangeable. The two chromosomes are going to have they're going to have a lot of the same properties, the same. But you couldn't state that as a rule of nature. There may be a difference, but I mean, it isn't left and right. We're not going to think that way. We're thinking about we have one chromosome here in that territory and one chromosome here, right?
Mm-hmm.
Now it could be that there are some differences depending on what these things are next to, and there could be a lot of complications, but let's just not consider it. We'll consider them the same, that they have the same chance of popping out. Yeah. But now if the variation occurs in this transcriptional area here, this part that tells you how much of that gene is going to be transcribed, or if it's going to be transcribed at all, okay, if the variation occurs there, then you're going to have a difference in how these two things express. Okay, so you could have have I entered an area of confusion? Okay, these little signals in front of the gene often tell you how much of this gene is going to transcribe, and whether it's going to transcribe at all. So
transcribing it can be, as in being passed on. Right?
No, turned into mRNA. Okay. Back up here.
Okay. Yeah.
Okay, back up here. A really important, two real important terms you have to learn really well is transcription. It goes from DNA into RNA or messenger RNA, and translation goes from messenger RNA into proteins. So that's probably the most important note that you have to take, and the most important thing in order to keep track of these conversations. Okay.
All right.
One big takeaway. So I'm talking about how much transcribes. We could end up with 7 quarts of protein here, this one, but we have a slight difference in the transcriptional area here, so we only get 3 quarts here of that one, right? So altogether, we're going to get 10 quarts of the big A, okay, basically, but because the gene is the same, well, this is going to be a little a because that is a variation. I think I have confused. I've gone too far, haven't I, Andrew?
Maybe a little bit.
Yeah, a little bit. Okay. So table all that. Don't worry about that. But the big takeaway here is the two terms heterozygous, meaning you have 2 different genes at that location between your mama and your papa. Yeah. And homozygous, meaning that your mama and your papa are cousins and they have the same gene.
Yeah.
Okay, well, they don't have to be cousins. They could just they could well have the same gene. But anyway, so that's important. And the other third important takeaway is to try to understand this difference that inside of one individual you can have 2 genes and no more. So when SFT starts talking about created heterozygosity with Adam and Eve, they can only each have 2 variants at the most. They could have Adam could have an uppercase A and a lowercase a, and Eve could have an uppercase B and a lowercase b. So you can have 4 variants. So that is the most created heterozygosity you can have for that locus. Okay.
Yep.
So, but within a population, you can have a great deal of variation. We can have, up to 1,000 of them, so we can have a ton of different.
A, B, C, D, E, F, G. Yeah.
B1, B2, B3, B55. So you could have a lot. And then tracking how, the thing is that even though these genes vary, you're going to find that they pretty much produce functional proteins most of the time. And if they don't produce functional proteins, you're going to have an individual that either varies in a trait or is somewhat weaker or sicker than another person or dies entirely. The thing is, you don't have to worry about genes that don't function, because those organisms die, and they most often die before they even turn into a zygote, or before they even before there's any fertilization at all. So that is what they call purifying selection. That if something really goes wrong, it'll kill you. and you'll never end up in the population. So a population will end up. Let's see if I can add another page here, and then we're going to stop and we're going to kind of review and ask questions. But you know, you got all bloody hell. So you got all these individuals in a population, okay, and we're looking at a population, and you can have all of this variation between those two genes, and some of these guys might have white fur and some might have red fur and, you know, black fur, you know, depending on which genes they have in the population.
Or something like big antlers or curved antlers versus like if we're talking about like deer, long Great Dane legs or little dachshund legs, you know, depending. By the way, Andrew, there's a really interesting bit of information about why some breeds of dogs have short legs.
I might have a good relevant question now. So you got that population, and like you're talking about all those different variants. So if you have within that population a certain variant, like long legs, for instance, and there's there's more of those, then obviously that gets transferred, continues on into the next generation, right? In other words, so that's kind of how it.
Well, more likely to, because there's more likelihood of mating, right?
Right, and there's more of that gene to be passed on, right? It's more likely that gene that's going to survive and continue, and
right? Since sex is a stochastic process mostly, it's statistical in nature. So like Speed said, it's more likely to continue. It doesn't always, because of things like genetic drift and things like that, but we won't get into that, right, Speed?
Yeah, we got to continue, but so okay.
So but we are talking about populations here. You know, we have a population, so we have these two yellow genes here. But what if a glacial dam breaks and there's a river put between? Oh, I'm sorry, I did it wrong.
Then it's not gonna
Okay, so there's a river.
There's the environment just affected it, right?
Right. So now, this yellow gene, or having that yellow gene, is a little more popular, and it's not non-existent in this population, right? And so over the next million years, because that river isn't going to just go away, you know, and they can't cross the river, these two populations of insects are going to speciate, because this gene may end up, you know, this gene could drift out. You know, it could by accident. Well, not with this setup, okay? Because, well, it could. It could drift out of the population, or it could fix in the population. And if it fixes in the population, then we're going to have lots of these little guys that are all yellow, right? So they're going to be pure yellow. So it depends on what happens. So that's the stochastic nature of evolution. But we got a speciation event here where all of a sudden we got red-furred buffalo and we have black-furred buffalo, okay? And then, of course, they will continue to mutate in other ways, and they'll continue to diverge from each other because there's a separation, that sort of thing, okay? But now it's possible that, and backing up, okay. It's possible that this population doesn't separate and these yellow genes drift out of the population naturally because they just didn't, and that's what they call genetic drift. It's also possible that they somehow fix in the population for some reason.
Can you elaborate a little more on that? Drifting hasn't so.
I can provide an example or analogy.
Yeah, please do that.
It's not a perfect analogy, but I like it. So one kind of example of genetic drift is what's called a bottleneck effect. You've probably heard that somewhere, but basically it's so say you have, in this case, a bottle of different kinds of candies or marbles, for instance. Let's just say marbles, and they're different colors. So in this case, the different colors represent the different alleles, the different gene variants in the population. And you're going to shake the bottle, and you're going to get a random sample from that when you let some out. So it's not going to represent the whole population of marbles in this case. So that's basically how one example of genetic drift, where you're basically taking a random sample and letting that continue onward. Does that make sense?
Yeah.
Yep. Yep.
Yeah. It could just it could happen that, you know, for some reason these guys here die, right? And now we have a bottleneck, and now it's almost the same as a river, you know, flowing between the two. You're going to end up with a bottleneck. If the river cuts everybody off on this side, then you're going to end up with more yellow genes. Okay, and they might end up popular. On the other hand, this guy could die right here, and then we only have one, and that it just may happen that over time he doesn't end up with any offspring. We only have offspring from these guys. Okay.
I got something to add. Like, this is a perfect example of why it's so complicated, and there's so many different variants that this is exactly why we have so much diversity, right?
Yeah.
And why it branches. Exactly why explains exactly why it's a branch or a bush rather than right.
Yep.
Am I? Okay. All right.
Now another thing that we could explain here. Somebody was going to say something.
No, keep going.
Okay. For some reason, like, okay, this guy's dead. This guy's an idiot. This guy has no social skills. He's confused. All these reasons why they don't mate, and this guy gets crushed by a rock, so he's dead. So these guys are all out. These guys are out, right? Or they have offspring, but their offspring are confused, dead, or no social skills, or whatever. But anyway, this guy's offspring ends up in the future. All of his offspring, right? So he becomes like the source of that gene in the entire population. So it's possible that, even though there were tons of things before him, right? He had a daddy and a granddaddy and, you know, a grandma and all that stuff. There were tons of things before him. He has this mutation, but his mutation just happens to be the one mutation that makes it into the future of every member of that species. So we have like a mitochondrial Eve or a chromosomal atom, and that happens because of that stochasticity, you know, of I mean, anything could happen. So and oddly enough, stochastic. Okay, oddly enough, we see that a hell of a lot. You know, if you go back like 200,000 years in a population that is not really, really huge, you'll find that these genes came from one individual. You know, well, that is how one mutation ends up in the future, because that mutation is unique to this guy right here, right? Something went wrong in his copying of the genes, and he ended up with a mutant, and he just happened to be the one that was the granddaddy of all, that sort of thing.
Yep.
Because all the other branches died out.
Are we gonna go over a little bit of how mutations work today, Speed, or would that be some other session?
A little bit. I think what we want to look at is condensation of DNA, maybe.
Okay.
Here's an administrative question there, Speed. Do you want to go on for another 5 or so minutes and then take a break, or do you just want? Are we planning on going more than an hour?
Oh, we're almost at an hour already. No, we're about 45 minutes.
Yeah, we're at about 45 minutes, but.
So do we want to take a break? I don't know what that means.
Kick back, talk about something else. Everybody
go to
the restroom, whatever.
Oh yeah.
I'm just considering, you know, if there might be brain overload, or you could spend 5 minutes, you know, doing a review of, you know, the last hour, and then, you know, take a break, and then at the top of the hour, everybody reassemble.
Okay. Are you sensing brain overload?
No, I'm just suggesting it.
Okay.
How are you feeling, David?
Okay, to me, well, no, I'm fine. To me, the only thing I would add would be I have to be probably the most lay person that you could introduce this to, right?
Good.
And to me, it makes sense. I mean, it's easily sensible to me. Like, and that's what kind of baffles me about these creationists, why they think that this is all bullshit. I just don't understand, because as a complete layperson, and what science has studied and come up with, it makes sense to me. So
yeah,
I don't know
if I gallop through really fast and use all these terms without explaining any of them to you, transcription, translation, heterozygous, homozygous, loci, whatever, it's obvious how you can become swayed by the rhetoric, right? I mean, it's like, created heterozygosity. Okay, well, here's why created heterozygosity can't work, because you can only have 4 in Adam and Eve, right? At that one locus. But he'll say, oh no, there's all these other variations out here of genes, right? But what doesn't happen, okay, it does happen, but it doesn't quite happen. I don't know how to say this, Andrew, but I mean, genes do duplicate and move around a little bit, but for the most part, you're not going to have a gene up here. Okay, say I've got the uppercase A, lowercase a, uppercase B, lowercase b. If I've got a C up here, it's not going to end up down here in this locus, right? Not normally, yeah. I mean, some weird evolving things can happen later on, but I mean, you can't count on that, and it's very, very rare, that sort of thing. So anyway, that's why the created heterozygosity thing doesn't make a lot of sense.
and SFD, if you're listening, if it doesn't make any sense, how about you start making it make sense?
Yeah.
Making an actual model.
And there you go. And that's what I mean. I mean, from what little bit that I know, it makes sense to me. And all the crap that I've ever listened to with Sal and with anybody else that has tried to explain any of this stuff, none of this stuff that they talk about makes any sense whatsoever. Like, so is that my bias, or is that because I already believe evolution is true, or is that because of the actual information that's being?
Yeah,
well, it's probably a combination of both. You can never get rid of bias, to be honest.
Right. No, I absolutely agree. And that's why I was asking you guys' opinion. Would that be, for a person like me, is that my? It truly doesn't. It truly, like, I don't know. It makes sense to me. I can follow along the actual true science of it, but I don't know. Like, for instance, a guy like Sal, like the stuff that he talks about in a high, an educated person like himself, I just don't understand why he. I mean, I guess it's just because it goes against his religious beliefs, and that's it.
Right.
But I have nothing in the game, like in this instance, right? Whether evolution is true or not, it doesn't matter. It's not going to make any difference on whether I believe in a God or not, to me. Like, so I don't have any stake in the game, I guess, but he does. Does that make sense? I don't know.
Yeah. Well, they came up with a little fix for created heterozygosity, and they said, okay, fine. Adam has, uppercase A, lowercase a, but Eve has B and BB, but she has multiple zygotes. So God created all these different genomes inside of Eve. So God repeatedly raped Eve and gave her all these different different combinations of eggs inside of her when he created her. That's one thing they did on the SFT channel a few months back, which is really interesting.
Except, but the problem with that, wouldn't that be because like only 2 zygotes make an offspring? Like, so how did the rest get incorporated?
Yeah, at most Eve could have 40 kids, so if she had 40 kids, right, which is they have actually said that out loud that she had like 40 kids, then she could put 80 combinations or 80 variations into the future, right? If every single one of those were different, but then of course they would combine with Adam over here, and some of those before you got to the ark would drift out. So you may end up with like 50 combinations, but then when you got on the ark, you got 6 people that are breeding when they came off the ark, because I guess Moses was like over the hill and they didn't do it anymore. So so you could at most have 12 after you get off the ark. So that is their created heterozygosity maximum. They screwed themselves with that ark story. They should just drop that, and they might get away with. But still, Eve could just put 80 out into the population, and then we have 6,000 years for this gene to mutate, which is a pretty rare event for a gene to mutate in any one generation, right? I mean, somebody's going to mutate, but whether they get into the future. But then in 6,000 years, we have to have this massive variation that we find in our population today. It doesn't work. So
see, and that's just all made up crap, right? Has there ever been
this idea
in the history of humans that a woman has actually had 40 children? Has that ever happened? I don't think so.
Well, and the other thing is that there's absolutely no woman that has multiple genomes inside of her. Where do women store their eggs? I forget.
Ovaries.
In the ovaries.
Yeah, ovaries. Okay.
Yeah.
So I mean, there's no way. So they just made this up. So this is called ad hoc, and it doesn't say that in the Bible anywhere. It doesn't say that God had sex with Eve multiple times, and that's how I view it. I mean, that's the only way you get zygotes into,
Yeah.
I don't know. I don't know what the hell he did.
We're not here to talk about creationist genetic speed. We're here to talk about actual.
I know. Well, I think using creationist genetics a little bit helps.
Yeah.
Illuminate.
I understand.
Right. Okay. So back to the beginning. So we got a cell. Inside the nucleus, we have chromosomes in their territories. And these chromosomes are sort of like they're in a state of being being loose and being compacted. And I'm going to show a picture, about what compacted is. And then when it comes time for the cell to divide, these things compact up tremendously into those little X-looking chromosomes that everybody believes chromosomes look like, right? But they don't exist that way in the nucleus normally. So we have transcription, and then we have translation to proteins, that sort of thing.
I mean, think about it this way, David. If they were in that X shape, how would they get transcribed? How would they be open enough for? This may be getting ahead, but like, how would they be open enough for the gene to be transcribed into a protein? Like nothing could attach there because they're all condensed and coiled up. Does that kind of make sense?
Yeah.
Yes. Yeah. That absolutely does. Yeah.
So here's the other part. Okay, so you've got the double-stranded DNA, and those things wrap around something called histones. And histones consist of, I think, 9 proteins. But you don't have to worry about that. Just call it a histone. It's like a particle. It looks like a little barrel. And 192 of these bases here, each one of these is called a base, basically. You can't see my cursor. So, those little bars connecting the double strand, each one of those is a nucleotide, okay? And 192 of those wrap around that histone, and then there's about 50 in between, so it looks like beads on a string, okay? So you got 50 in between.
Yep.
And those beads can, depending on epigenetics, can slide along that string, or they can basically lock that string up so that it can't be transcribed. Okay. But there's little areas in between where the transcription happens. It's like string connecting beads on, beads. And every bead has a wrap of 192 bases around it, and it has about 50 in the middle. So there's so there's a string connecting the beads. Well, then they can coil up further inside of the nucleus in an operational cell. Okay, an operational cell as opposed to one that's dividing. They can curl up a little bit, and they can make it very difficult for any transcription of that gene to happen. So that gene is essentially kind of cut off, right? And then when the cell divides, they further condense into these hyperstructures, and then they eventually end up in that little X at the bottom. And then it's ready for mitosis, ready to do division, that sort of thing.
Okay.
So that's mitosis. And then there is meiosis, which we should probably save for another day, because that's where you enter, recombination enters the picture. And you learn a little bit more about genetic variation inside of, in what they call diploid. Diploid means we have 2 copies of each chromosome. So diploid sexually reproducing organisms have a tremendous of opportunities to create variations in their offspring. So if you have 100 children or 40 children like Eve, they'll all look different. Okay.
Yeah.
Yep.
So, but there's a lot of reasons for that, and that has to do with meiosis, not mitosis. So that's kind of a different thing. But anyway, so this is how DNA compacts. And when they talk about epigenetics, most of what's happening is called remodeling of these genes. And then, let's see, where do they mention? This is a really good picture about mitosis and meiosis, but we'll save that. I'm trying to find let's see, there's a meiotic divisions. Here's a little blurb that I copied from, I should mention the guy's video. This is where how DNA, okay, eight of these histones group together to form a unit called the nucleosome. And then the DNA wraps around the helix wraps around that thing like a solenoid. And then they wrap further to form supercoils, and it gets more complicated. So,
how is this, Andrew, for you? Is this too basic for you, or is this interesting, or is it?
I mean, some of the stuff I didn't know, but most of it I already knew.
Yeah. Well, I figured that, but
here's a nice little picture of these guys wrapped around the histones, so you can see the beads on a string. And has Demeter complained yet?
I don't think so.
You need to be careful.
Oh, you gotta be careful.
I have a question for Andrew, if you don't mind. How much of this did you learn in high school, and how much of it did you learn on your own?
Most of this actually I learned throughout high school, but some of it I did learn on my own. Yeah,
because I don't remember learning anything about genes.
They didn't
in the 1970s.
Yeah, when I went to school, they didn't even know half this shit. So they taught us. They taught us that there was a cell and there was some junk inside the cell, and then there was a nucleus, and that was it. Yeah, it's like that's what we know. You don't know anything else.
It's amazing to know the history of what we've learned about stuff is pretty awesome.
Yep.
Very cool.
This may be a little off topic, but like a little brain break, basically. But like, someone actually responded to my LPP comment from yesterday.
Oh, did they? What'd they say?
Let's pull that up and read it.
Okay. Oh, you sure? I don't derail the conversation.
We can go back. We can go back.
Yeah, we're taking a break. Taking a break.
Sure.
Oh, Demeter actually wrote something. He put everything. Apparently, he's okay with everything. That's good.
God damn.
We need an auditor of this course.
Yeah, we do.
Yeah.
Y'all didn't do Punnett squares in high school biology? Nope. Punnett squares? I don't even. Yeah, I guess they did know what Punnett squares were back then, but no, they didn't go that far.
Is that dominant recessive kind of stuff
Yes.
like Mendel stuff?
Yeah, Mendel's genetics.
We might have done some of that.
That's where you figure out how those combinations get into the offspring, basically.
Well, and I know we did in college in the 80s.
I hate that this is not popular enough, like, for our audience, I guess.
Yeah.
Well, it is Sunday morning on Father's Day too, so.
Yeah, that's definitely. Yeah, that doesn't help. That's for sure. Yeah.
Well, what you got to do is you got to put all of this information together, and you got to have funny little pictures in between, and you got to make it light and humorous, and put pictures.
Yeah.
I don't know.
Yeah.
And of course,
on that note, happy Father's Day to everyone on the panel and everyone out there.
Thank you, Andrew.
Thank you.
Yep. Yeah. So, I think that probably is a wrap for one day. What do you think?
Sounds good.
So
I'd like to keep going. Do you guys want to keep not now? I'm saying, but I mean, future.
Yeah.
Future.
Oh yeah, definitely.
Yeah, I think the next place to go is basically this mitosis and meiosis, basically the differences and maybe how recombination works, that sort of thing.
Yeah, we could also throw in a little bit about mutations here and there.
Yeah, we could go through mutations. Let's see.
I think Benghazi thought this was going to be a fashion discussion. Damn. Talk about dashed expectations.
Well, I got some jeans on. They're kind of ripped up, but I could
jeans started with miners. They were miners in late 1800s needed tough pants, so they started wearing jeans.
Well, I think cowboys liked them too.
Well, actually, no. At first, cowboys didn't wear them. They didn't want to be associated with the miners that were wearing them.
Oh, okay.
It wasn't they didn't get associated with cowboys until much later. Cowboys would wear regular pants and then chaps over them, like leather.
That's right. Because they go into like some of these old mines and find these Levi jeans, right, that are worth like tons of money.
Yeah, and they're still holding up.
Yeah.
So people would be mining without pants. Is this the Herman? Was that the early
pants with an extra?
Yeah, they would change. Right, they would change and leave behind all kinds of jeans in these mines, abandoned mines, and these people go in and search the mines and find them, and they're worth a ton of money, I guess.
So you can take the old jeans and then use make a pattern from them. To reproduce them.
So the area of Minnesota that I come from is where the iron mines were. And when I was a little kid, I talked to an old man who worked in some of the older mines. Like when I was a kid, they were open pit mines. But when he came over from the old country, is what he called it, they were all underground mines. And what they would do is they would have a man with a pick crawl into these caves, go down this shaft. And they would crawl into these little crawl spaces, and they would pick away until they could plant dynamite. And then they would all run out and blast it, and then they would go down and clean out the rubble, and they mined the ore that way. And when he came from the old country, he wanted to go to work as a miner. So he went to the mine office, right, and for the underground mine, and asked for a job. And they said, okay, go and wait by the elevator shaft. And when a body comes up, you got a job.
Holy crap.
Yep.
Oh, wow.
And that's how unions got started.
Wow.
Because after a while, people said, wait a minute. These people died at a pretty good clip, I guess.
Yeah.
Well,
they often prefer children because they can fit in a smaller mine shaft.
Oh, yeah. Yeah, children are great. Yeah. Tinier the better. Toddlers are perfect.
Yeah.
This channel does not
and then when they
child labor.
And then when they died, that was lunch, right? That's how atheists were born, because
Oh yeah,
children, right? And that's how they started eating children, right?
But anyway, when one of the miners died, they probably took the genes off and stored them away for later on. That's why I think they find genes down there. I don't know.
Yeah,
don't know if that's good. But, yep.
Yeah, we're getting into some deep stuff now.
If we're gonna talk about some other stuff, should we take this thing or this screen now?
Did you want to put up your thing?
Put up the comments.
Oh,
the comment thing?
You sure?
Yeah. Why not?
Yeah, I'll probably divide this and maybe edit this a little bit. I'm not sure what I'm going to do with it, and put it up as content for teaching this stuff, and I don't know, refine it is what I would like to do, and put up a duplicate video, and then we can just take this one wherever you guys want to go. So, Father's Day special, whatever. We talk about Amy's stream last night. There was a lady on there. What the hell was her name? Something
Jennifer,
about
Church of Entropy.
Yeah. Yep.
Yeah.
What do you know about this person?
Oh, she debated T-Jump on MDD. First of all, for anybody that did see Amy's stream last night, I want to apologize.
For Leo?
For me.
Oh, what did you do?
If you caught that part.
Give me a time. Decide if I forgive you.
No, I came. Well, I had a little bit too much rum last night, and I came on there, and I couldn't hold it in and couldn't hit the mute button before I ended up subjecting everybody to the sound of my hurling. So I apologize for that.
Boy.
Oh, no.
Yeah.
I didn't see that part.
That's one thing.
Yeah. So if you want to see that, go check it out. If you don't want to see it, skip over that.
Wait a minute, you actually threw up live on Amy's channel last night? Is that?
Yeah. I feel so bad.
The camera on you?
Yeah.
What's the timestamp?
Boy.
I'll have to check again. See if I can get it to you.
That's
So
okay. You don't have to do that.
Well, just so you know,
I came on right after Will.
probably not your proudest moment, but just so you know, I am an AA member. If you ever want to talk.
Okay. Yeah. I don't think I really have a problem drinking. I just, I know.
Yeah.
Basically, what happened was that my friend didn't want me to take the bottle in the car with me when he took me home, so I took a huge swill. Didn't realize.
Yeah, I've done that. Yeah.
But anyway, you're talking about Jennifer from Church of Entropy,
right?
Yep.
So
yeah, she's kind of like Deepak Chopra.
Yeah.
She claims to have a physics degree, but she says things that make absolutely no sense if you understand physics or anything.
Yeah.
She's a nice lady, a nice gal, but I don't.
And she's bright.
She says some crazy stuff.
She's definitely bright, but she has some sort of essence thing. I think was the source of consciousness or something like that. Or...
No, she thinks that consciousness is a quantum photon quantum waveform.
Yeah. Wait a minute. Entangled? Was it entangled or was it?
Yeah, something like that. I don't think she even understands what she's saying.
Yeah. Yep. Yeah,
And
that's problematic. But
she claims she has a theory of everything, right? Wasn't that her claim?
Oh,
yeah, I think so. And she understands consciousness.
Yeah.
She made a prediction. So in T-Jump's debate, in her debate with T-Jump, she said that your consciousness has to leave your brain while you're sleeping. The reason that you need sleep is to keep your brain from electrocuting itself. So your consciousness has to leave your brain in order for it to rest. And she predicted that she said you can find the consciousness in this sort of donut shape around the brain. And T-Jump's like, great, there's something we can test. And she's like, well, no, it would be unethical. We can't do that.
Unethical?
Yeah, because you would have to interrupt somebody's consciousness in order to test it.
So I have a donut floating around my head?
When you sleep. your consciousness
When I
leaves your
sleep.
brain.
When I sleep.
I'd pull a Homer Simpson. I'd pull a Homer Simpson and eat it.
Now, what about animals? Does she ever mention that? Do animals
Oh,
have
yeah.
that too? Because
Yeah,
all
no,
animals
not just animals,
sleep too.
everything.
Yeah. Everything.
Everything. Because
yeah,
she called T-Jump a brain having supremacist for denying that things without brains can have consciousness.
Oh my god.
Oh.
So I came in when Leo was being very rude and very disruptive. So I was immediately sympathetic to her. So I went back and listened to not a lot, but probably about one fourth of or even half of the way through. And she sounded bright, had a lot of stuff, but then every now and then I could hear the woo coming out, with the S and stuff and things like that. So I was bracing for that, but I listened to her sympathetically, and I was kind of appalled at the way she was treated at some point. And as far as the over talking stuff, I mean, the over talking shit has got to stop.
Wow.
Yeah, she'd only get a couple words out. Like she'd try to start saying something. She'd get three words out, wouldn't even finish a sentence, and people were hopping all over it. Yeah, it was pretty bad.
Yeah, and that's I'm going to become less tolerant of that shit as I go on this channel. I think, as far as putting people in the back room, I got to grow a fucking backbone.
Yeah.
That is a hard thing to regulate because
it is, especially when your friends are around, people you like.
Right, and you want to give everybody a chance to say something, but
Like,
people have
Leo
to have to let somebody say something. Yeah.
Leo got mad at me yesterday because I put him and I think Cerberus in the back room for a moment so I could get a word in edgewise and calm them down because they were just running over each other, and it's like, stop it,
Yeah.
Because even with Amy's stream, when that conversation happened with Leo, I had everything I could do to keep myself from shutting the stream off and going to look at volcanoes in Iceland, So, I mean, nobody's going to watch that shit, So if we actually want people to watch our content, so if you go on a stream and you do that to somebody's stream, you are really being. It's like going into somebody's house and taking a shit on their living room floor,
I've told Leo, sometimes he'll be right, and he'll be arguing with somebody who's not actually arguing against what he's saying, but because Leo doesn't hear them.
Yeah. There's been streams like that on here, right? This not for quite some time.
Lots of them.
There's been a few, and I found that watching those streams, if I was watching and not actually a participant, I had to shut it off. I'd have to say, okay, I have to walk away from it because you just can't stand the screaming back and forth. It's just gibberish. So it's just, what's the point?
And then if you're trying to get a transcript, there's no way you can read the transcript. They can't even transcribe that shit. So it just screws everything up.
And I think that's why doing this kind of stream with genetics and phylogeny and stuff like that is good speed, because it adds more content to your channel, in my opinion.
Yeah.
Right.
Yeah. With a small room, just a few people, so that there isn't a lot of going in many different directions. So I don't know.
Yeah, well, that gets to be a problem when you have too many people on screen, and especially when you have one person in one position and then 6 people on the opposite side of them. Then you have everybody trying to correct them at once, and
it's not to be offensive, Speed, but sometimes that is why I leave the streams, because there are so many people in that it's unlikely I'm gonna myself get a word in. So
well, I am finally tuned to you saying Speed in the background in that low voice.
I appreciate that.
But you also got to force your way in a little bit too. I'm like you too. I will sit back and not really say anything, but there's many times where I don't really have anything to add anyway. But if you have something to add, but even then, sometimes I've found where I've wanted to add something, but the conversation is going so well that you don't want to sidetrack it too. So I'll just keep my mouth shut, and you try to remember what you want to, and then the conversation ends up, going into a completely different subject when you wanted to add something to it.
Exactly.
You find that happening all the time. So,
and then you have people. You've got people like Batman who will talk for 5 minutes straight and then get mad at you for interrupting them and talking over them. It's like you can't just sit here and filibuster and then get mad that we talk over you. You got to stop at some point and let somebody talk.
Right. And that's why anytime I try to add anything, I try to make it quick. You try to say something quick, and then I try to do that too.
Yeah, I don't want to
being on the streams like this has definitely helped me practice my brevity.
I don't want to be offensive, Andrew, but you come in and then everybody's like talking about something, and then all of a sudden I got dead air, and I look and you fucking left the stream, and that was going to be your turn. And you're like a jackrabbit as far you're in and you're out, you're gone. It's just like,
I'm sorry.
Settle down.
Yes,
okay.
Andrew,
I'll try to
definitely,
do better.
please push your way into the conversation more. You need to definitely do that, because usually what you're providing is the scientific conversation that this channel wants, right? So that's why we want you to do that. So
that's why
you don't have to be Darth Dawkins, but you can definitely be assertive.
Okay. So we've had a couple of on weather reports. We've had a couple of things where we've gone in a 1.5 hour fight about physics with people, and I sort of tune out because I don't know enough physics to be boldly stating opinions, right? So I don't know if I like those conversations.
My mentality personally is where I don't know something, I don't speak about it.
Yeah.
Like when LPP, for instance, posts his semantics arguments and stuff like that, I just don't really care because I can't say anything.
That's something we ought to talk about at some point, is this business of information in biology, because LPP mentions this Royal Society issue in 2016, where there's about 15 papers on information, semantics, pragmatics, and syntax and all that in biology. And not one of those people ever mention intelligent. They might mention intelligent design in one or two of the papers, but none of them agree with it. They only push back on it, right? So that's where you got all this bullshit about semantics and pragmatics and all of that. And it's
Nephilim Free does the same thing. I'd definitely be interested in that discussion.
Yeah, we could go over some of those Royal Society papers. I've read most of them twice, and I got them highlighted, but it was like a year ago. And then I've got a ton of books on bioinformatics, and I've even got the Creationists. The Creationists got together in, I think, 1986 or something, or maybe, I don't know, maybe it was 96, and they did an entire huge book. They basically rented a room in some hotel, and they called it some big convention. And all the ideas and creationists got together and talked about information. And Sal Cordova went over that book. I've got it sitting here somewhere. Where the fuck is it? But I've got a whole stack of books on that sort of stuff, and I like talking about it. I like talking about information as physical. My idea of how information is entirely physical. There is no woo component to information at all.
Yeah.
I think I make a case for that.
Yeah. Speaking of LPP, I don't think he ever. You remember LPP.
Where's that comment? Low PP.
Same comment from yesterday. Someone just replied. SpongeBob Imagination.
Who the hell is that? Is that SFT?
I don't know.
Yeah.
Really
whoever that is, that's a dip. Yeah, he's a dipshit.
But he sounds kind of like SFT a lot of times. I don't know.
People have accused him or that person for being SFT, but I don't think it is. And only because I think SFT has used that SpongeBob character and stuff in the past. So I think that's the only reason why they think it is him, but I don't think it is.
Yeah.
Different person.
We're trying to find your comment. Oh, there you are. Andrew coming. Okay, there are 2 replies. Okay, so.
Oh yeah, one was I can't tell if because the flame person I'm friends with online, we often see each other in these debates, and like we're just like, yeah, we're getting some entertainment out of this. But I don't know if he was responding to my comment or SpongeBob's comment.
Said that was perfect. Let's see. So SpongeBob said, trusting the scientific consensus for no reason beyond the fact that it is the scientific consensus is a terrible strategy when it comes to unproven and unverifiable claims.
Which, by the way, isn't what I was arguing.
Yeah. So, like, I trust...
Yeah, it's a pretty long comment. Oh, go ahead.
I always trust the consensus, like, when I bring my car into the shop, because I don't understand modern engines. I trust the consensus of the mechanics there. I don't know. Just, I mean...
Right, yeah, that's kind of similar to the point I was trying to get across.
Yeah, like, if you're not an expert...
I'm not knowledgeable. Yeah.
Okay, so science is littered with so in the last 20 years, in the last 40 years, what have they gotten wrong, really wrong?
Right. That was the point.
There.
Right. What was that?
That was the point that Danny, when we were discussing this yesterday, me, you, Danny, and Andrew, right? We were discussing this yesterday about consensus, and that's true. I mean, how often have they gotten things wrong? So it's a rare thing. It's generally they get things right. So
yeah, and if you look back into the 18th and 19th century, they got a lot of shit wrong, right? But we weren't exactly a technological race back then. You know, we were just poking our heads out of the.
We also didn't have the scrutinous process of reviewing each other's papers and stuff like that as much as we have today.
Right.
And I think, right? I think that like nowadays you've got computer models, right? Which is huge. So, you got this huge computer power that helps it along. So you can really advance far more.
Yeah,
and it's less likely, it's less likely a computer's going to get something wrong, right?
And they love this
calculation and that. Yeah.
Yeah, and the other thing is we have the internet now, and there's just a lot of self-checking going on and a lot of communication. Yeah. Anyway, this Planck's principle, he came up with that in 1930 or something like that. So
yeah, Max Planck.
It's Planck.
Is it Planck?
Yeah, Planck.
Yeah,
Planck. I call him Planck. Imagine a historian. Oh, and because I speak proper English. Oh, geez, thank you for that. The fuck? Jesus Christ, you just put me off, friend. Living 1,000 years in the future decides to review all the scientific consensus. And what are they going to find that's wrong? Probably nothing. Some things about cosmology and some things about particle physics, I'm sure, because physics is where the big revolutions really happen.
Right.
But in biology, I don't know.
I mean, my 2 big complaints with his comment is that, for one thing, the fact that science gets things wrong and learns from that isn't a bug of the system. It's what it's supposed to do in the first place. And the second thing is, like we were saying earlier, I wonder how he would respond to the fact that Newton was wrong in the sense he's saying, but we're still using the physics he came up with today, kind of highlights another thing, which is science isn't often wrong as much as incomplete. Like I like to, it's like Russian dolls, basically.
And the alternative they're suggesting is that we trust creation science, which starts with the Bible, right?
Yeah.
Right.
Yeah, exactly.
I mean,
okay.
go over to Sal's channel and listen to any one of his streams and listen to all the biblical passages he intermixes with his so-called science. And you know that he is biased as hell.
It's funny too that they complain that science is you can't trust science because it changes, but the creation model has also changed. They used to most creationists, pretty much all of them used to be species fixes. They thought that all species were created at creation. And now they've gone to this idea of kinds. That's not in the Bible, and that's not what they used to believe.
If it hadn't been for somebody standing up and saying, "Hey, how did all those species fit on the ark?" they would have kept talking about it that way. They would have never changed. So they only changed to accommodate what science is pushing back with.
Right.
And then,
I mean, I like this analogy that a professor gave me once through email. Basically, science learns and progresses. And if you're saying consensus is wrong, therefore it's not trustworthy now, it's kind of like saying, kind of like how a business manager keeps doing the same strategy over and over again, but obviously it's not working. So why should we trust him and do business with him if he's not learning from his mistakes? So that's kind of the thing science does. It learns from its mistakes. And it's funny how what they forget to point out is the people who are proving them wrong, or to be more specific, incomplete, and finding the holes in our knowledge are the scientists. So scientists prove other scientists, or you know what I mean? Like,
yeah.
And if we look back to 2021 from 2121, and we look at cosmology, and we chuckle about what are we going to chuckle about? Most of the physicists today are disagreeing with one another, and they have different models, and they freely admit we don't fucking know. We don't know this. We don't know that. We don't know what dark matter is. I mean, nobody's claiming dark matter is a fucking beanie baby from another dimension.
Yeah.
Nobody's making strong claims about the stuff they don't know. So I think science is in pretty good shape now, as opposed to where it may have been 200 years ago.
Right.
But even then, those people argued with one another and admitted when they didn't know something. I mean,
right. Yeah. I mean, it's
they weren't idiots.
I think the strong thing about science isn't so much that it gets things right, is that when it gets things wrong, everyone harpers on it. And like you see a paper out there in the literature, most of the time when it gets something wrong, all the researchers are going to start shotgunning it and saying, this is why it's wrong. Here's how to fix it. Go back and retest your results and things like that. And the problem gets fixed or it gets resolved. So,
another thing is that I don't know has it ever happened where there was a scientific belief and then it was disproven and then later on it was reaccepted? Because that's essentially what they're trying to have happen with creationism, right? Or young Earth or anything like that.
Not really in that sense, in the strictest sense.
Because that would be like us disproving oxygen and then going back to phlogiston.
Yeah.
Well, if you look back at what Lawrence Krauss, I think, was kind of famous for saying is scientists, they want to prove each other wrong because that's what makes you famous, right? If you prove that something and, you come up with something better or something that improves the scientific theory, the consensus, if you prove that wrong, you're going to be famous, and that's what they want to do. So they're all they're striving for that.
Right. You learn the most by being wrong, not by confirming the status quo.
Yep.
And this thing here, truly, no one has a stronger faith-based belief than the atheist when it comes to scientific consensus belief that the wholly naturalistic abiogenesis, free from intervention, guidance, and direction, occurred on a long ago unobserved Earth. Well, for myself, basically, I will admit that I have faith in complexity and chemistry. So there's that. But I don't have any alternative because I don't know where this intervention, guidance, and direction could have come from. And I haven't seen anything plausible that could do that other than complexity and chemistry. And I've looked into a lot of individual little tiny edge cases in abiogenesis, and they have a ton of ideas and possibilities. And so far, I'm not really doing that on faith because I don't know how it happened. And I haven't picked a method yet. And I'm open to them showing me what this thing is that did intervention, guidance, and direction, because in my life, I've never had an example of God's little fingers coming in and stirring my oil and vinegar in such a way as to create wine. I've never seen it. So I don't know, is that faith or is that just sort of an agnosticism that I have? You know, I don't know how it happened. Yeah. But I mean, everywhere I look, like Sal always talks about eukaryotic evolution. Well, go Google that. How did eukaryotes evolve? And there's just paper after, I'm quoting SFT, paper after paper after paper on it. And with all sorts of, possibilities and mechanisms. And then you look a little further and you find out that these coesins and things that are on the surface of the nucleus, that they have homologous genes that go all the way back to prokaryotes that didn't have anything like that. And then you find out that various kinds of endocytosis use some of the same proteins that the nucleus uses. And then you go out and find out that these nuclear localization signals are actually in prokaryotes, which didn't have a fucking nucleus. So they were used for something entirely different, yet they're there, so you can see footprints going all the way back into the past. There's something up here, and it's, and that's why I have this faith-based belief, or is it just a healthy agnosticism? I don't know.
I mean, how have we actually observed cells absorbing other cells and then the cell within the cell survives? Like, has that been a thing that we've actually observed?
I don't know of a paper that shows that, but quite possibly. There's, and they like to bounce all over them. They really have turned their focus. Like when I first got into this community 2 or 3 years ago, it was primarily evolution that they were attacking. And since last fall, summer and fall in 2020, they started attacking abiogenesis more, right?
Origins or bust.
Yeah.
But Speed, I don't know if you want to save it or not, but you're going to find some good material on Sal's last video that he did last night.
Okay.
I don't know if you want to do it now or if you'd rather save it for the weather report, but there's some pretty good stuff on there.
Yeah, I want a bookmark.
Random Ray video that y'all were talking about.
Yeah.
I want to do bookmarks on it, because if we do it now, it'll be almost impossible to find the good parts and get the whole gist of it, right?
Yeah.
But yeah, I've been keeping an eye on Sal Cordova.
Yeah, there's one section in there. Like I said, he went off on a rage, and you're gonna find some good material in there for sure.
Okay, cool.
So
do it on the weather report. What day is it? Sunday?
Yeah, today's Sunday. Yeah.
God, it was just Saturday.
Yeah, yesterday I think.
That's the way the calendar works.
Wasn't that yesterday? Yeah.
That always surprises me. I'm sorry. I'm surprised every day. I wake up and I say, well, how the fuck did that happen?
Yeah. So Sal, his grand plan is to attack evolution, right?
Yeah.
Maybe I'm sure you've heard him say this, but he seems to think that he wants to win this battle, right? He wants to win the battle against abiogenesis, evolution, and then he's going to move on to geology and prove.
Oh boy, I can't wait till he gets there.
Yeah, so he's going to move on to that, and then the granddaddy of them all is physics. Then he's going to attack physics to prove. Yeah. So he truly thinks this, like he's going to win this battle in his lifetime. He's going to overturn. That's overturning every scientific thing that exists.
Yeah.
Yep. I can't wait till
the
we get
delusion of grandeur.
Like, seriously, that's pretty delusional, I think. Wow.
Can't wait till he gets to geology, because he is cramming 700,000 geologic years into every year. And I really like to see how he's going to explain the rock formations in Washington that I've been watching.
Dapper
Dino. Sorry, go ahead.
What's that?
Dapper Dino said something in a debate last night that I thought was really, really quotable. Actually, I don't know if he got it from somewhere else or if he came up with it. He said the difference between young earthers and flat earthers is how long we've known they've been wrong.
Yeah, I was watching when he said that. That's pretty awesome.
Yeah, it's pretty cool. Dapper Dino's debate with Jungle Jargon was very informative. Also, Dapper Dino really presented a good case, and then so it was a lot of really good scientific information interleaved with the absolute comedy of Nicholas Whitmire saying really weird things.
Yeah, well, he essentially admitted that he had no idea what a dinosaur is. So how do you even?
But he knew that birds didn't come from dinosaurs.
Well, no, he said he knew that the birds are not dinosaurs, but he doesn't know what a dinosaur is.
He's just happy he got to say Jesus Christ a couple of times, talk about the gospel and everything. So that's all he wants. He doesn't care if he's ridiculously wrong.
How did that debate even come about? Like, I'm shocked that he would even do that, Nicholas.
I know. He came on my channel a while ago, and he came in just raging. He was gonna take me on evolution. And I got done with him in about 10 minutes, and I was trying to find the escape hatch.
Yeah. Yeah, he's out there, that's for sure.
He really is out there.
Yep.
That conversation I had with Tony on Smokey's channel about abiogenesis, that was just holy shit. Why did I do that? Why did I think I could talk to these people? I don't know.
Who'd you say, Tony? You just said
Tony, Tony. Yeah, that was where they got the thing that I didn't read all my books, that sort of shit.
Yeah. You don't see him that much, like he doesn't, or at least I don't.
Not as much. He has not been as active in the last month or a couple of months.
If you remember going way back, probably a year ago, remember he was like Smokey's right hand man when it came to science. Yep. Remember that, right? That's back basically when what you're talking about back then.
Yeah, it's interesting. Okay, should we shoot it in the head?
Sure.
It's up to you, boss.
Yeah, I think we should shoot it in the head.
Sorry, Randolph, what you said.
Okay, I think I'm going to edit this, I think, and try to organize that content so it's more, so it has some pictures of the notes and the things that you should take away: transcription, translation, heterozygous, homozygous, that sort of thing. And hopefully, David, we can move on from there. Yeah. And if you have anything, when you're thinking about this stuff, anything that's not clear, and when we dig into the transcription a little bit, that's where it might be a good idea for me to give a really short introduction to structural organic chemistry, so that you can read those diagrams that you see on the wiki, and so you know what ethanol is. Oh, and by the way, where did Leo last? Well, let's talk about this backstage. Okay, I'm going to end the broadcast.
Yep.
But I have a question about ethanol.
I want to talk about that too.
Yeah, let's see here. What the hell? What's happening? Okay, we're ending, guys. Thanks, everyone out there. I think